I'm happy to introduce Mark Lieber, who's he is a new to UT associate professor at the Marine Science Institute, which is in Port Aransas, if you're not familiar. The whole, just as sort of a background, if you're not familiar, some people aren't. So the Marine Sciences Department at UT is almost entirely, except for me and one other person, based in Port Aransas. There's an research institute there. It's a really great facility. You should check it out. So Mark did his bachelor's at Boston University, and he did a master's in the marine biology program at Boston University at Woods Hole, through Woods Hole. And he did his PhD with one of my favorite people in science, Andreas Teskey, at the University of North Carolina in Chapel Hill. He's published around 75 papers in his, I would say, fairly early career, which is very impressive. I was very excited for him to come to UT because his interests are very much aligned with mine. I'm almost like scarily close, until we worked in the same places. We've never worked together, but we're after the same interests, and that are sort of understanding the role of microbes in carbon cycling and through time, on the planet. So he uses just very briefly, he'll tell you what he's doing, but I'm excited because he's really sort of pushing the boundaries in terms of understanding what microbial communities that aren't cultured are doing in the environment. And so I'm sure he'll tell you about that. So go ahead, Mark.
All right. Thanks a lot, Brett. Yeah, today I'll be talking about the potential importance of diffuse hydrothermal fluids in sustaining life in seismically active deep sea environments. And I'll explain to you what I mean by that as I go along. And also, just in advance, if you have any questions that you don't get answered during the Q&A afterwards, feel free to email me anytime, or just happy to be in touch and build a network with you. Yeah. So let's see, something's not moving. Here's the outline of my talk. I'll briefly talk about the kind of work I do and the questions I'm most interested in relating to microbial energy cycling. Then I'll give you an overview of past research on a type of hydrothermal system that we call that have so-called focused hydrothermal fluid flow, and talk about how those systems are different from a much less studied system, which are the diffuse hydrothermal systems, which were the subject of a recent expedition I was on in December of last year. And I'll be presenting you some of the work and the plans relating to these diffuse hydrothermal systems. So my research, as Brett already explained, it relates to the carbon cycle on Earth, and more specifically, the role of microorganisms in controlling the cycling of carbon. And here's just a very general graph that shows you both natural and human transformations of carbon in the biosphere. And I'm mainly looking at not what's happening in the water or on land, but what's happening in the sediments that underlie aquatic ecosystems, and looking at the fate of biological building blocks in the long term. And so what factors determine whether the organic carbon that's produced in the biosphere is preserved in the long term and becomes part of a carbon sink, a vast sedimentary carbon sink, and what determines whether the organic carbon gets broken down and released as greenhouse gases such as CO2 and methane. I study these questions in mainly sedimentary environments, ranging from coastal sediments to open ocean sediments. That includes deep subsea floor sediments that are can only be reached by drilling hundreds of meters or kilometers below the seafloor using big drilling vessels. I've also done work on lake sediments, and that was more related to human influence on carbon burial. And also I've looked at Earth's crust and mantle environments and subglacial streams, and done some aquaculture related stuff. Today's work will be mainly relating to Earth's crust and mantle and sedimentary ecosystems and how those interact. So the key question of my research is what determines the fate of organic carbon in the biosphere. And this is a fundamental question because organic matter is produced on land and in the water column, and a certain percentage, oftentimes less than 1% of that is deposited at the seafloor. And then one of the biggest questions of organic geochemistry and marine microbiology, at least in sediments, is what determines whether that organic carbon is buried and becomes part of that geologic carbon reservoir, organic carbon reservoir that has huge importance in terms of driving atmospheric oxygen concentrations in the long run, and what determines whether microbes are actually able to access that organic carbon and gain energy from it, and then ultimately convert it to CO2 or methane, which are greenhouse gases that can be released back to the hydrosphere and atmosphere. And in order to tackle this question, I use a mixed bag of approaches. So one angle I'm focused on a lot is the study of microbial community structure, looking at bacteria and archaea, which are the two main groups of microorganisms in the environment, and identifying which ones are there and how are they structured in relation to environmental gradients. Another angle that I've taken is to measure rates of transformation processes using isotopically labeled substrates, and also looking at the genetic potential of microorganisms. So what kinds of genes do they have? Which enzymes do those encode? And which carbon transformation reactions are catalyzed by those enzymes? And lastly, an important component then is to not only look at the microbes, but also to look at what are they transforming, and that's the organic carbon pools in the sediments. What is the chemical composition of the organic matter? How does that change over time as a result of microbial community structure and microbial activities? So this combined then provides us with a better idea of what are the mechanisms that control the organic carbon cycling. And many of these methods can be used to be also quantitative, so we can get at quantities at which rates are taking place and which organic compounds are being transformed over time. And another kind of side project that's always been kind of, since my dissertation, been kind of looming in the background is what is the influence not just of that photosynthetic organic matter, but also what role do crustal and mantle environments play in perhaps altering the chemical composition of organic carbon deeper in the sediments. So I'm going to start out now giving you just a brief overview of how microbial food chains operate. So the typical microbial food chain that most of us are well familiar with from school is based on the production of photosynthesis of biomass by photosynthesis. So in the open ocean, that would be microalgae in a lot of places, such as phytoplankton, that produces organic carbon. And that organic carbon then can be eaten and taken up by different organisms to produce a food chain. The energy is coming from light. And then over time, as the organic matter as the organisms die or produce waste, some of that organic carbon finds its way to the seafloor, and that's typically in particulate form, where this organic carbon is present as polymers, such as carbohydrates, proteins, nucleic acids, and lipids. Then once that organic carbon is in the sediments, there can be a whole sequence of biological transformation reactions, and I'm not going to go into too much detail into those, but one critical one is the initial extracellular hydrolysis that makes these large macromolecules accessible to microorganisms, so microbes convert them using extracellular enzymes into monomers that they can actually take up and then gain energy from. And then there's a whole sequence of reactions that then in the end produce CO2 and methane as final products of this organic matter transformation chain, with different microorganisms being involved in the different reactions here, which I'm not going to go into, but listed here. You can see primary and secondary fermentation, respiration, and cytogenesis. And these are performed by different groups of highly specialized microorganisms. Now, there's the way ecosystems in nature work is not only through biological transformation reactions, and that includes the carbon cycle. So there is also a whole set of non-biological reactions that can strongly influence the fate of organic carbon. And those are shown here. So one involves the heating, so high temperature settings that can be found, for instance, in some sedimentary environments can catalyze the breakdown or thermocatalysis of organic matter to smaller building blocks. In the surface ocean, we can have UV light contribute to the degradation of organic matter. And then also in really deep sediments or environments where we have deep fluid inputs, we can have abiotic or geogenic reactions having a strong impact. And I'm not going to talk much about photodegradation, which is not relevant in deep subsurface sediments or the deep sea, but I'll be talking about the other two processes. So the first one being the thermocatalysis, which is heat-driven breakdown of organic polymers into monomers, and this high temperature can then also catalyze a number of reactions that are very similar to the biological fermentation reactions, can also result in the breakdown of monomers to the same end products or of fermentation and respiration reactions. Abiotic geogenic reactions are different in that these are actually rock-powered typically or mineral-driven, so they involve the reactions of minerals or inorganic rocks or with water, and they're independent of photosynthesis-derived energy. And one example is serpentinization, where we can have water reacting with reduced iron minerals in the Earth's mantle, and that can release iron oxides and hydrogen, which is a prime microbial energy substrate. That hydrogen then can continue to react with CO2 to produce methane or other low molecular organic compounds such as ethanol or methanol as alcohols, or organic acids such as formate and acetate. And all of these are also microbial energy substrates. In addition, we can have volcanic activity contributing hydrogen and methane and other volatile compounds. And something that I won't talk about today that occurs even at low temperatures everywhere in marine sediments is the radiolytic decay of uranium and thorium and potassium. And that can split, for instance, water into hydrogen and oxygen, and that hydrogen and oxygen can both then be used by microorganisms. So oftentimes when we study processes that are happening in deep sediments, and we're thinking of geothermal influence, we're actually studying systems where it's the temperature that is geothermal in origin, but it's actually photosynthesis-derived organic matter that is broken down by high temperatures in the sediments. And in these settings, we don't have the generation of organic matter by other processes, but it's still basically the photosynthesis-derived organic carbon that's being chemically modified at high temperature. Very different is the abiotic or geogenic actual production or synthesis of organic carbon, which is a reversal of this process where we have the synthesis of more complex organic molecules from CO2 and methane and hydrogen. And this can result even in the formation of monomers and even polymers, but that's, I'm not going to take it that far, and that gets very theoretical very fast. So what we then have in these environments where we have significant input of these chemical energy sources producing organic and inorganic compounds, then we can have chemosynthetic biomass production. So we can have entire food chains that are independent of photosynthetic organic matter, or largely independent of that, and mainly gaining energy that's ultimately produced by rock or mineral water reactions. And oftentimes when we talk about chemosynthetic biomass production, we also include temperature-driven breakdown products such as hydrogen and acetate that are ultimately of photosynthetic origin, but are the same compounds that can also produce by abiotic and geogenic reactions. So oftentimes when you talk about chemosynthetic biomass production, we're lumping both of these processes together. That's the general intro I want to give you, and then I'm going to now talk about give you examples of some hydrothermal systems where we have focused fluid flow being the main source of energy to microorganisms. And when I talk about focused fluid flow, this is a very, let's see, semi-quantitative statement. I'm talking about areas with fast advection, but what does fast advection mean? It means maybe advection on the anywhere from meters per day or meters per minute, but not much slower than that. And usually this channelized or focused fluid flow occurs through defined fissures or conduits. So it's very localized in terms of where the fluid flow is happening. And here are some examples of these types of systems. The first ones are hydrothermal vents, and then two others that are really important are seep environments where fluids are seeping through the sediments through local channels, and also mud volcanoes, which are larger structures where it's not just water, but it's also water with suspended particles, so muds that are being transported up to the seafloor. And the last example are subsurface oceanic crustal environments, which also have very highly channelized or focused fluid flow. These are by no means very well studied systems, but they are the best studied hydrothermal systems that we have in marine environments, and they can be typically found in very particular locations of the oceans. For instance, hydrothermal vents are very oftentimes located in areas where oceanic plates are being pulled apart. So we have seafloor spreading centers with a shallow magma chamber, and that shallow magma chamber drives a very steep temperature gradient to the seafloor. And as a result of that high temperature gradient, water that is in the sediments gets heated up, and gases are also released, and that results in the flow of low-density fluids from the subsurface to the seafloor. Now, as those fluids are being transported up, they're also drawing seawater in from the sides, and that seawater then reacts with these magmatic fluids and then releases, as a result of that, large quantities of hydrogen and reduced metals such as manganese 2 and iron 2 and ammonium and methane and various reduced sulfur compounds to the seafloor. While the temperatures often in the actual hydrothermal vents are very, very high, oftentimes too high for microbial life or any form of life, at least as we know it, can be very rich oases of life then at the seafloor. And these types of oases of life can be microbially dominated. So in the actual hydrothermal chimneys, where we have a lot of hydrogen, we can have strict anaerobes, such as certain methane-producing archaea. And I'm listing here Methanopyrus, which is a genus that has one species called Methanopyrus candleri, which is the organism that grows at the highest temperature known so far, which is 122 degrees Celsius. This organism uses hydrogen from these fluids and bicarbonate from seawater to produce methane, but also sulfate reducers can be very important, such as members of the thermodesulfobacterium group, which use hydrogen and sulfate in order to gain energy. We can also get macrofauna in very high abundances. So here we have Avinella tubeworms, which grow at, or can actually survive temperatures, not grow, but survive temperatures over 100 degrees Celsius for short time periods. And these have chemosymbiosis with sulfur-reducing bacteria in their tissue, in their gill tissue, and they are able to, the bacteria are able to gain energy from reducing the elemental sulfur, and the Avinella tubeworms are then basically living off the biomass of the bacteria and utilizing oxygen that's present in seawater for their respiration. Other examples are these shrimp, Rimicaris shrimp, which have sulfur oxidizing bacteria associated with their their tissues and all kinds of other symbiosis, including ones that are still very poorly understood, such as these ciliates here, which are unicellular organisms of the genus Folliculinopsis, which have unknown symbionts and can form these beautiful purple films that cover hydrothermal vent environments. Now, as I mentioned before, hydrothermal vents are typically found in seafloor spreading centers, so they're highly localized. The seafloor, and the red dots here show all the known hydrothermal vent environments, and yellow dots are the ones which are unconfirmed, and there's been several hundred that have been discovered so far. The other type of focused fluid flow system includes seeps and mud volcanoes. And seeps include both hot seeps and cold seeps, and mud volcanoes are similar systems, but just larger and expunging rather than just water and fluids. They are also transporting sediments. These seep and mud volcano environments are shown in blue here, and those are very oftentimes found in subduction zones, not only, but that explains why they're geographically separated from hydrothermal vent environments. And they're very interesting environments to study from a microbiological standpoint, and from many other angles too, in that they are areas where we have energy-rich fluids being transported through the sediments. So these are sediment covered parts of the seafloor, and in some areas where we have in close proximity to hydrothermal vents, we can also have hot fluids going through the sediments. So these are the so-called hot seeps. And then we have these other so-called cold seeps and mud volcanoes that mainly occur in subduction zones, where we have one plate going under the other. And then as a result of water reacting with the mantle rock that is present in these environments. So we have sediment-rich in water reacting with mantle rock in these environments that can release hydrogen and methane and organic acids. And those same compounds can also be released in these hot seep environments. These environments are very spectacular, oftentimes to look at from the seafloor, in that they can host a very high biomass of bacteria, so microbial mats. I'll show you some photos in a minute that live there, but they can also have macrofauna, so many species, or not many, but a few species of clams and mussels that can live in these environments. And this high biomass of microbiota and fauna at the seafloor is supported largely by methane, which comes from below. So that methane that is being transported up to the seafloor is the ultimate driver of this very high biomass in many of these systems. And the way this works is that very often we have methane coming up, then coming into contact with sulfate from seawater. And when methane and sulfate are together at high concentrations, this can stimulate the anaerobic oxidation of methane coupled to sulfate reduction. And we can have these consortia here of archaea that are shown in red, and bacteria, and these are sulfate-reducing bacteria in green, that together oxidize the methane. And the fluxes of methane can be so high that we can have a very, very high biomass of these methane-oxidizing archaea and sulfate-reducing bacteria occurring in these systems. Then that production of, or that oxidation of methane and coupled to sulfate reduction produces huge amounts of hydrogen sulfide, which can then be transported up to the seafloor. And that's ultimately what then supports the vast biomass that we at the seafloor. So right at the seafloor, where oxygen and nitrate are present, we can get certain bacteria that are specialized in the chemoautotrophic processes involving the oxidation of hydrogen sulfide using oxygen or nitrate from seawater. And that can generate a lot of energy and explains the high biomass of these microbial mats. These microbial mats are typically dominant in areas where there's a very high flux of hydrogen sulfide, in areas where the fluxes are a bit lower and the hydrogen sulfide concentrations are not as high. We can also get macrofauna, and macrofauna cannot simply not tolerate the same levels of hydrogen sulfide as these bacteria can. And we can get these clams and mussels that have sulfur oxidizing or sulfide oxidizing bacteria living with them in their tissue as endosymbionts. And therefore, we can have a very high biomass also of macrofauna in these systems. And it's basically these microbial mats and macrofauna that enable us to find these seep environments when we look at the seafloor. So very oftentimes we'll have the deep sea will look largely devoid of life or only have minimal biomass of organisms, but then when we get to these seep environments, we suddenly find these gorgeous microbial biofilms or microbial mats that cover the seafloor, and we can also find high biomass of these bathyomodulus mussels. We can also even have the formation of carbonate reefs in places where we have very high rates of methane oxidation, because anaerobic methane oxidation can then trigger the precipitation of carbonate and result in hard rock structures in otherwise soft-bodied sediments. And some of these hard rocks or carbonate structures that are produced can then also support a very high diversity of all kinds of macrofauna, including sponges and cold water corals, and also even octopus and many fish species. Now, one of the questions that we want to address in our research is to understand what drives these big differences in the communities of organisms. And one thing that's clear here is we have very big difference in macrofaunal communities, but I'm mainly looking at the microorganisms. And I've looked at a particular environment, which is a hot seep environment, where we have steep temperature gradients, and areas where we have fluid flow from below, and also areas where we have steep temperature gradients without fluid flow from below. And the interesting question here was to see, it's usually difficult to disentangle the impacts of temperature and fluid flow on microbial biomass and microbial community structure, but this particular environment in Guaymas Basin allows us to do both, because we have both types of systems in close vicinity. So here's a particular sketch that I drew to exemplify this. We have some areas with steep temperature gradients of up to 80 degrees at a few meters below the seafloor, but there's no fluid flow at these sediments. We call these the non-seep area. And then there's another environment which is much better studied, which Brett has done a lot of research on and even discovered new phyla of bacteria and archaea, and which is the seep area, where we have strong flow of fluids that are driven by geothermal activity in deeper layers and supply large amounts of methane and organic acids and hydrogen and also petroleum compounds to the seafloor. And this is what we call the seep area. So this is, as I mentioned, in Guaymas Basin. These two areas are geographically largely separated, and they look very different when we look at them from above. So the seep area has these beautiful microbial mats at the seafloor, and the areas without the seepage, which gets hot several meters below the seafloor, looks like just normal deep sea sediment with traces of life, but not a lot of biomass of microorganisms or macrofauna. And one of the interesting findings, and this is mostly work done by my former PhD student, Lorenzo Lagostina, and it was published a couple of years ago in Communications Biology, was when we tried to disentangle the impact of temperature and fluid flow or energy flux on microbial communities, we saw some clear similarities and differences depending on which taxonomic level we were looking at. One interesting thing we observed was that the relative abundances of bacteria to archaea followed very similar patterns when we looked at these hot areas, hot sediments without seepage and hot sediments with seepage. And those are shown in green and red here. And you can see they fall along a similar line here, which indicates that the ratio of bacteria to archaea on the y-axis very much follows temperature. So what we found here more specifically was that sediments with a temperature of less than 40 degrees Celsius had ratios of bacteria to archaea that were greater than 1:1, suggesting bacteria were dominating of archaea. And this is based on gene copy numbers of 16S ribosomal RNA genes, whereas sediments where the temperatures were above 40 degrees had almost everywhere a strong dominance of archaea over bacteria, or ratios of bacteria to archaea below 1. Now, this is something that may not surprise some of us who've studied microbiology, basic microbiology textbooks, where it's oftentimes written that archaea are so-called extremophiles that dominate only in extreme environments. These are outdated textbooks, but it's still written in many of them. And therefore, archaea dominate over bacteria at high temperatures. However, when we look at the environment, there have been only very few studies that have shown that to be the case. This conclusion that archaea dominate at high temperatures is mainly based on pure culture studies, where people have taken samples from the environment and added substrates and grown the microbes in the laboratory. And there they found that most of the microorganisms that they could grow successfully in the laboratory above 80 degrees Celsius were archaea. This, however, was the first study where it was shown that there really is this temperature relationship, and it seems to be independent on whether there's fluid flow or not. Fluid flow does, of course, as one would expect, also have a strong influence, because it strongly impacts the amount of energy that is transported to these microbial communities. And here we looked at the bacterial and archaeal community structure at the phylum level using non-metric dimensional scaling method. And we could see there's clear influence of temperature, as we already saw earlier, that low temperature samples are in blue here, clustered together. But we could also see then that the non-seep area, where there's no fluid flow, has a very different community fingerprint at the phylum level in bacteria than the seep sediments, where we have this strong flow of hydrothermal fluids from below. We can see the same thing for archaea, that at elevated temperatures, we have clearly different archaeal communities at the phylum level. And we can also look at other taxonomic levels, such as the zero operational taxonomic unit level, which is oftentimes used as a proxy for the species level. And here we also see very big differences and clear separation of bacterial and archaeal communities between at the species level, between the hot sediments with and without and with seepage. One thing to note, when I've now talked a lot about these seep environments, which are very interesting and similar to the ones I'll be presenting to you later, is that in most seep environments, we have a strong, because the fluids have been highly channelized and flown through hundreds of meters of sediments, there's a strong interaction between the fluids and the organic matter that is in the sediments. And in places like Guaymas Basin, especially where we have very steep temperature gradients, we have a lot of thermal or temperature-driven breakdown of organic carbon in deeper high-temperature layers, which then supplies large amounts of hydrogen and methane and organic acids that are actually originally of photosynthetic origin back to the seafloor. So even though we see this chemosynthetic biomass production and stimulation of microbial life by in this system in a seep environment, most of the energy that's actually reaching the seafloor was originally produced by photosynthesis. And although at Guaymas Basin, we probably also have input of deeper fluids that contain abiotically produced hydrogen and methane and organic acids, that signal is so weak that we almost basically can't see it. And it's really driven by these heat-driven thermogenic processes that are provide photosynthetic organic matter. With that, I'll now talk about the last part, the subsurface oceanic crustal environments, which are another type of focused fluid flow systems. And they're very interesting in that, so the oceanic crust covers roughly 60% of the seafloor. And that's wherever we don't have the terrestrial plates, we have oceanic plates, and that excludes the continental margins, but all the abyssal plains are sediments have oceanic crust underneath. And these oceanic crustal environments are also they are full of life and in many locations. And there's a wealth of microbial processes that take place in them. And one of the drivers of microbial life in these oceanic crustal environments is actually related to seawater that is drawn in places where we have seamounts that are protruding above the sediment layer, and water is drawn into these seamounts, then gets heated by geothermal heating, and then is expunged at other seamount environments where, as a result of the lower density of the fluids of the water. And due to the water entering here, we have then the potential for water-rock reactions to support life and provide a system where we can actually have rock-powered energy or energy that's really driven, that is potentially photosynthesis independent, supporting life. Now, these are very difficult environments to study because very oftentimes we have to drill through hundreds of meters of sediment in order to be able to reach these crustal aquifers, and then we have to drill into very hard rocks that can break open as we drill into them, and it's a very challenging place to study microbiology, also because of the contamination risk. The only way that it's being done so far is using drill ships. So here at the top you can see the JORDIS Resolution, which is the American drill ship of the International Integrated Ocean Drilling Program. And one popular way of studying subsea for life with this drill ship has been by installing so-called borehole observatories, which are in essence a pipe that reaches all the way through the sediments to the seafloor, where it has this plug, is a reentry cone for the coring device, but also a plug, which is called the cork or circulation obviation retrokit. And these cork observatories or borehole observatories then have pipe that goes down through the sediments where it's sealed, and then into the oceanic crust where it's then open, and then allows crustal fluids to flow into the borehole, where they can then be rather easily sampled in order to study the chemistry of the oceanic crust. So these borehole observatories can be, for instance, placed strategically in different parts of the flow path of these crustal fluids to look at how is the crustal fluid being transformed chemically as it is transported or traveling. And the other way we can study life and processes in oceanic crust is by actually doing coring. So in this case, we can get rock cores by drilling and then transport these up onto the deck and then do some analysis on them. One challenge with rock coring is that all the rocks that have been brought up have been heavily contaminated with seawater and drilling fluids, which can themselves sometimes be very high in DNA. And so it can be very challenging to study these rocks. So we have to do stringent decontamination procedures where we sterilize the entire exterior and then try to crack them open and study what's in there without introducing any contaminants. Because that's so challenging, it's been preferred method, or that is probably most studies have involved, has been using these borehole observatories. And one interesting finding that has emerged from studies of borehole observatories is that seawater-derived organic matter and oxygen and nitrate seem to be very important in sustaining the microbes that are oftentimes found in these borehole observatories. So it almost suggests that there's not much potential for a real for microbial truly, let's say, rock powered microbial community, but actually most of the life in the oceanic crust is supported by organic matter from seawater and seawater-derived electron acceptors. Now, this is an interesting finding, and it can be explained with different, there are several explanations for that. One potential explanation is that when boreholes are driven, they typically will then, the fluids inside will be largely dominated by large cracks in the oceanic crust where we have most of the seawater flow, most of the water flowing through. And because of the high rates of water flow we can get significant amounts of organic matter from seawater and also oxygen, even reaching locations that are kilometers away from the entry points. However, that's based on my past research, that's not the only place where or there are certain disadvantages even of these borehole observatories. And those are, for one, we're mainly looking at the borehole chemistry is mainly controlled by fissures with high fluid flow. And also we're mainly looking at the planktonic microbial community. So the organisms that are living in the water that is circulating through the basalt. But we know from sediments and also the continental crust that most microorganisms are not living in the fluids, but actually living in biofilms or attached to rocks and particles that are getting exposed to these circulating fluids. So because of that, I then took a different approach quite some time ago and looked at these rock samples, and particularly ones that had very small fissures, micro fissures that were only a few micrometers or tens of micrometers wide, where we didn't have much fluid flow, but enough fluid flow to result in chemical reactions between the water and the surrounding rock. So we can, and those chemical reactions result in this visual alteration of the rock to produce these black halos. And I then specifically looked at the microbes that are living within these veins inside the rocks. And using a combination of DNA-based methods and also isotopic analysis of sulfur and carbon, we're able to show there's clear correlations between sulfate reduction by bacteria and the distribution of genes that are indicative of microbial sulfate reduction. And also there were some clear patterns between the detection of genes that have been linked, that we could link to methane production and methane oxidation, and the isotopic compositions of carbonates and organic carbon in the rocks. So with that suggested to us, and the isotopic compositions here were clearly different from those of organic matter from seawater, which then led us to propose that it was actually in these very fine veins that are away from the major fluid flow channels in the oceanic crust. We have perhaps the release of microbial energy substrates by water rock reactions. And in this case, we have a lot of olivine in the particular rock formation that we were studying. And the idea then was proposed that olivine reacts with the water and releases hydrogen, organic acids, and ultimately perhaps even methane. Now, I've talked a lot about these focused hydrothermal fluid systems. And since there's not much research to talk about diffuse systems, I'm going to be making a lot of comparisons and then presenting you some hypotheses for future research that my group will hopefully be testing over the coming years. But before I get into that, diffuse hydrothermal fluids or diffuse fluid flow are, again, vaguely defined areas where we have slow seepage through the sediments. And in this case, I'm guessing, or based on my conversations with hydrogeologists on ships, we're talking more on the level of maybe meters per year, maybe even less than meters per year that's taking place, where we have very, very slow seepage of deeply sourced fluids, perhaps from the Earth's mantle, through the sediments, through hundreds of meters of sediments, all the way to the seafloor. And these are the least studied and least understood hydrothermal systems, and their importance to subsea for life at this stage remains rather hypothetical. Just to give you an example of how little is known, so there've been over 300 hydrothermal systems discovered so far. Almost all of them were on ridge axis or in subduction zones, and I'm only aware of 3 or a few more hydrothermal systems that are with diffuse flow. Yet it's been estimated that between 50 to 90% of the global hydrothermal fluid circulation is happening in these sediment-covered diffuse systems and not at the better studied places with more rapid fluid flow. It's also been estimated that roughly 70% of hydrothermal heat flux occurs in these diffuse systems. So from a planetary standpoint, they could be very important on regulating the exchanges of the chemical exchanges and heat exchanges between the Earth's interior and Earth's surface. Yet we don't know much about this diffuse hydrothermal flow on seafloor and subseafloor life. And these systems probably don't occur anywhere in the seafloor. There are large parts of the world's seafloor where sediment layers are very thick and impermeable to fluids, so there's probably no fluid flow. But they do occur in a lot of places. And this map here shows you in the white lines are areas where we have plate boundaries, and that's where we find hydrothermal vents and most cold seeps and mud volcanoes. And then those black lines that you can see everywhere, these are the systems where we expect to see many of these diffuse hydrothermal systems. What these are transform faults, which are areas that are within the spreading centers where we have high activity, hydrothermal activity, and then also we have these and seismic activity, and also then these black lines that can go for thousands of kilometers away from these spreading centers, which are known as fracture zones, which are traditionally believed to be seismically inactive, but research of the past decades has shown that not to be the case. And the way these diffuse hydrothermal systems may work is illustrated in this plot here. And this is based on a transform fault where we have the spreading areas of a spreading center here on the left, where plates are being pulled apart here on the left-hand side and on the right-hand side, and then connecting these cracks that are where we have the spreading, we have the transform faults where the plates are actually rubbing against each other as they're being pulled apart. These transform fault zones are locations that can have very high earthquake activities or seismicity, and whenever we have this seismic activity, this can result in the fracturing of the oceanic crust, and also can result in fractures in the sediments and produce conduits for fluids then to be transported up from below, especially when we have pressure. When plates are rubbing against each other, this can result in tremendous pressure, which can then expel fluids from deep down here all the way potentially to the seafloor. Or there may also be areas where the same action can draw down fluids from the bottom of the ocean into the Earth's mantle, where that water can then react with the mantle rock to produce hydrogen and methane and other organic substrates. So we know that for sure it is accepted among hydrogeologists, at least is my understanding, that these water exchanges are taking place. However, we have not found any evidence that stimulates life in these environments, and that's one thing we've set out to do. And it's not an easy thing to do, because these diffuse fluid flow systems occur over vast areas of the ocean floor, and are yet probably pretty localized in distribution, meaning they occur over areas of maybe square kilometers, which is rather small, of course, in these systems. But there have been recently some cruises that my group, also my former group at ETH Zurich, were involved in with colleagues from the GEOMAR, which have looked at a particular fracture zone that is between the Eurasian plate and the African or Nubian plate in the North Atlantic. And in this area, we have the Gloria Fault, which is a long fracture zone that's here between Spain and the Azores. And then there we have the Azores Plateau, which is an island. The Azores are an island archipelago that's made of volcanoes with also high seismic activity and fracturing of the oceanic crust in this environment. The first cruise took place in 2020 and was unsuccessful. Looked at all these places here in the Gloria Fault where there have been past very major earthquakes, so we know seismic activity is there. This is not an inactive location, but there are earthquakes and potential for fluid flow. However, there was no success. More recently, we looked at locations that are closer to the Azores, and this was just in this past December, in the hopes of finding evidence of deep mantle fluids reaching the seafloor. And what makes this location very attractive, in my opinion, is not just the high seismic activity, but also we are in an environment that is, these are abyssal sediments with very low water column primary production. So there's very little organic carbon coming from the overlying water column to the sediments. Sedimentary organic carbon content is an order of magnitude or more less or lower than in Guaymas Basin. So we have an environment where there's very little energy available from photosynthesis. We also don't have high temperatures. These are uniformly cold sediments, so we don't have much potential for thermogenic activation of ancient organic carbon in the sediment. So it's an environment, and a desert environment, ecological desert environment, where even the input of small amounts or of low fluid of containing energy substrates from the underlying mantle could potentially result in significant stimulation of microbial and macrofauna life in the seafloor. The expedition I embarked on with colleagues from Germany and Portugal was Meteor Expedition 186. I was from Lisbon to Gran Canaria, and so these are Portuguese and Spanish waters, and most of the time we spent looking around the Azores four areas with potential fluid flow. We had some days with great weather like this, but most days were not great weather. They looked more like this, where it was actually impossible to do any coring in most of the study area. So we had really tremendous waves that, in this case, we even had an underwater video camera in action at the time when we had these massive waves hitting the ship, making it very dangerous to retrieve our equipment. So that posed many challenges and meant that we couldn't do the same sampling program we had originally planned. Here's another 2 photos that just show you how much this ship, which is 100 meters in length, was really getting rocked and in motion due to the rough seas we're experiencing due to both a hurricane reaching us from the south and then shortly afterwards an Arctic storm hitting our region from the north. We also encountered damages with our equipment. Here, a line broke when our equipment was at 4 kilometers water depth and it had to be fixed. It was quite dangerous. But nonetheless, we also had some happy moments and we celebrated Christmas. And but now to the science. So because the weather was so bad, we had to spend out of the 5 weeks, 3 entire weeks just hiding between these 3 different islands here in the São Jorge Channel, next to the volcano of Pico, which is here, which gave us a beautiful view on clear days. And we were just hiding in here. And because we had nothing else to do, we started coring, not having much hope that this would be a place with fluid flow from below. However, we were fortunately proven wrong, and there's more data than I'll be able to show you, but some of it is not, I'm not allowed to show you yet. We did find some very interesting geochemical changes in the seafloor, so we could see that total alkalinity, which is a proxy for microbial activity, and also deep fluid sources, showed some very divergent patterns. So we had locations where total alkalinity was probably mainly driven by the organic matter mineralization in the sediments, resulting in this increase in dissolved inorganic carbon, driving this increase in total alkalinity, and the blue site here. And then we also had locations where total alkalinity was dropping greatly within the upper 4 meters of sediments. And this was something that we then thought could be a clear indication of some deep hydrothermal fluids reaching all the way to the seafloor. Some more recent analysis have now shown that our initial hunch was probably correct. We can see that the concentrations, for instance, of magnesium and calcium and boron are extremely different, especially with increasing depth at this location, where we suspect there to be deep hydrothermal fluid input. And based on the depletion of magnesium and increase in calcium, we even suspect that these could be serpentinite fluids of mantle origin that are reaching all the way up to the seafloor. So now the next question or challenge is to really show that that's the case. And what we're planning to do there, and this is a project I'm going to propose for a PhD student over the coming years. In my group, we'll be looking at the compositions of specific microbial energy substrates, looking at methane and hydrogen, short-chain organic acids and alcohols, aldehydes and carbon monoxide. These are all low molecular weight organic compounds that are and inorganic compounds that are known to be released by serpentinization. We'll also be looking at the isotopic compositions of some of these, and this will hopefully allow us to distinguish between sources that are where we have photosynthetic organic matter being the ultimate source, and geogenic or abiotic processes deep down in the Earth's mantle being the sources. And we know already now that photosynthesis-derived organic carbon has isotopic depletions in the -20 to -22 range typically, whereas abiotically produced organic compounds are typically much less isotopically depleted. And so therefore, we're hoping that we can use the isotopic compositions as a further proxy for that will help us identify the contribution of deep mantle-derived fluids in terms of the energy substrates to life. This will be combined with analysis on inert elements, so noble gases, where the ratio of helium isotopes can provide clear indications of mantle origin, and also strontium isotopes and many other dissolved chemicals will be analyzed by collaborators at the GeoMod to hopefully provide a clear indication of deep mantle-derived fluid origin. This will then be combined with microbial community analysis. So we have samples that are from areas with these strong fluid alterations, which we can compare to control sites where we have more typical deep sea sediments where photosynthetic organic matter is probably the main energy source to life. And we can look at microbial community size, see if there's any stimulation of life in areas of fluid flow. And we could also look at the community structure of microorganisms based on signature genes such as 16S ribosomal RNA genes, and also looking at specific marker genes of processes that might be stimulated by these deep fluids, such as anaerobic methane oxidation. We can also combine this with data or compare this to data that our collaborators will produce on macrofaunal biomass and community structure. And here it is entirely possible that in these desert type deep sea environments, even a low rate of subsurface fluid flow with microbial energy substrates could elevate the macrofaunal biomass significantly above that of surrounding deep sea sedimentary environments where there's no fluid flow. So this is something we're hoping to find, and we'll know more over the coming years. Lastly, being a largely microbiology lab, my project will also involve the enrichment of microorganisms from these sediments using hydrogen and methane as key substrates. And then this will hopefully enable us to not only based on the gene and genetic signatures that we find, propose which organisms are involved in the turnover of these substrates, but also show that using these enrichment assays, which we hope to combine with genomic analysis and Raman microscopic analysis. With a Raman microscope, we will then be able to directly look at associations between sedimentary matrices and these microorganisms that are involved in this, hopefully the cycling of hydrogen and methane. So with that, I think I'm out of time, but I've shown you the plans for the future, and I'd be very happy to take any questions you have. So thanks for your attention, and yeah, I guess I'll zoom out of the presentation.
Great talk, Mark. Super interdisciplinary, which is always great to see. Does anybody have any questions? Go ahead if you do.
Yeah, I'll ask one. So I'm curious if the prediction from the diffuse hydrothermal fluids would involve there being a whole lot more serpentine production than just, away from the tectonic boundaries. Is that the direction you're thinking of heading? We've under-predicted that potentially. I'm sorry, I didn't follow. I was saying, I'm just curious if you're predicting in the diffuse fluid flow areas, away from the tectonic boundaries, that there's this role with serpentization. Do you predict then that the process of serpentization in the middle of the ocean plates might be under-predicted? We're expecting that to happen hundreds of meters deeper, in part, maybe the crust, and also in the mantle rocks, or lower crust, where we have water coming into contact with these ultramafic rocks, and then there to be fluid flow upwards from these deep environments all the way to the seafloor. Now, one challenge is that we're talking about cold sediments, and one can ask, so why should the microbial stimulation be happening at the seafloor and not hundreds of meters below?
That's something we don't know yet. It would be much better to have a drilling expedition that is able to go all the way down there to maybe even areas or layers where the serpentinization is happening. But that's not something we can do. It's something we hope to do in the future. One thing to add here is the pH of these fluids is around 7.5 to 8.2. Past research in highly serpentinitic environments suggest in these environments we very oftentimes have very little life at pH ranges of 10 to 12. So it could also be that there isn't much life down there, but it could still be that the most of the microbial activity is somewhere in between in sediment layers where the pH is already approaching a more favorable regime for the microorganisms.
Yep. David, go ahead.
I might yield to Mark if he's got a more
pressing question. Go ahead, Mark.
No, David, go ahead. I'll ask after.
Okay, so what I was going to ask is, we are interested in the geysers on Enceladus, which have been suggested to maybe have hydrogen in them associated with some sort of something similar process. Are you aware of similar types of structures to those that you talked about today that are not basically in the deep ocean? That is, maybe they occur under an ice layer or possibly in like a large lake, something that's more easily why is everything out in the ocean?
Well,
it's hard to get at, like you said.
No, but I mean, I'm not a geologist, but my understanding, the ultramafic rock where serpentinization is happening only occurs in the oceanic environment. It's not something you would find as much in terrestrial settings. Are there better places? Well, the problem is when we go to better places that are more easy to reach, like Guaymas Basin, there we very oftentimes have very high input of organic matter from the overlying water, and we may have heat-driven breakdown of organic matter in the sediments, and that probably overprints any impact of these deep fluids. So I guess we can think of where would be the ideal place. I'm not aware of any shallow water settings that are ultra oligotrophic with very little organic carbon that are located in a fracture zone. Yeah, I'm not sure, but maybe somebody else has a better idea.
How about are there any under the North Polar ice caps that?
Yes, there are seep environments under the Antarctic and Arctic ice, I believe. I think there would again.
That doesn't make them easy to get at, of course.
Doesn't make them easy to get at, and also we oftentimes have high water column productivity there also. So the ideal setting is really organic poor, minimal life supported by organic matter from above.
Thanks.
Mark, did you have something?
Yeah, I was curious. You had this, schematic drawing where essentially you said the organic production happens where you have methane from below getting in touch with the sulfate from the ocean. And I was just curious, so, if there is flow going, for example, if there's upward flow, so the methane is dissolved or is it gaseous?
It's dissolved.
So if
largely, not only, but the methane that gets eaten is dissolved.
So I guess I was just wondering, so let's say you have an upward flow, right, that brings the methane up. How does the sulfate, does it just diffuse it against that, or does it sit in little dead end pores, or for that matter, if the flow is down, It's
a great question. So one thing is that the flow of fluids is not homogeneous. It's rather localized, and I'm not an expert on this, but it's been shown that hydrogen sulfide, and know that seawater also gets drawn in, and that's in part resulted in because of the heterogeneities in the fluid flow. Even microscale over centimeters, this can result in sulfate being drawn in some places. And
okay, and then it gets mixed by dispersion or diffusion. Okay.
Yes.
And is that same? So the the methane comes from what in your situation?
So in Guaymas Basin, most of it comes from, we have reached temperatures of several hundred degrees Celsius in an environment that has a very productive upwelling regime. And so we have very, very high organic carbon content, and that organic carbon then gets broken down. In other environments, serpentinitic systems, we do have methane also. And there it's not quite clear how it's generated, but it is clearly there. And people have proposed there have been different explanations. Some people have argued it's actually not methane that's produced there, but you have magmatic methane that is present in fluid inclusions. And that can then, as the serpentinization takes place, the rock basically falls apart and releases that methane. But there have also been, and people are arguing about that, whether there can also be other pathways, such as more Sabatier type reactions where hydrogen can react with CO2, perhaps catalyzed by certain metals that are in the rock, be transformed into methane. And temperature also, you can have very high temperatures down there. So you can also have high temperature reduction of CO2 with hydrogen.
You've mentioned the serpentinization of the rock falling apart. How important do you think is there's this sort of whole reactive cracking community that looks at how serpentinization sort of, although it has a volume increase, it creates stresses that essentially auto-brecciate the rock. How important do you think that is relative to just flow in pre-existing fractures? Or is that
upward flow?
Well, I guess in general, flow through the system. I
wouldn't know, but from the people I've talked to, the biggest factor are earthquakes in these environments that really create these fissures through the rocks, and then the water flows over larger areas through the sediments, which are rather permeable. But this is getting very interesting, but it's outside of my area of expertise.
Cool. Thanks.
All right. Thanks, Mark. That was awesome.
Thank you.
All right. See everybody later.
All right. Bye-bye.